My name is Sergej Filipov, or you call me Sergej. I'm a visiting Liverpool professor in the group of Vitaly Hutariansky, and my background is polymer physics. So in my work, I'm using a lot of physical instruments like dynamic light scattering, small angle X-ray, neutron scattering, zeta potential, electrophoretic mobility experiments, just to study polymers, any type of polymers, polymers for drug delivery, polymers in food science. So, and today I will be talking about one of such techniques. The name of this method probably you've heard before this talk. It's a dynamic light scattering, and I will address in my talk to the three, actually, it will be organized in three parts. I will give a very brief, very basic introduction to the method, and I will explain how this method could be applicable to the investigation of food science, and I will also share with you some hints how to make a decent dynamic light scattering experiment yeah, with your samples. So, just to start my lecture, I have to say that we live in a very random, in a very stochastic world, and this is general true because we never know for sure if there will be rain tomorrow or today. We know that the stock market is a subject of actually great fluctuations. uh, we we know, speaking from a broader point of view, that a lot of issues, a lot of well parameters around us is a subject of fluctuations, is a subject of randomness. For example, the global mean sea level is actually changing over years. The climate is a very random and very probabilistic issue. So if you look on the mean average temperature, you can see actually a lot of fluctuations over the last several decades. Uh, you can find the examples of stochasticity not only on Earth but also in the universe. For example, the number of sunspots is changing all the time, right? So this chart, I hope, will convince you that actually the spot number is actually also a very actually random and stochastic process. Of course, coming back from microscopic level to the, let's say, to the real world, to the world of human beings, we can find examples of stochasticity and randomness, let's say, if we're talking about ourselves. So this is my heart rate during one of the runs that I made in Kazan, in Russia. So you can compare my heart rate of this particular round with the heart rate actually another round that I made here in UK. And even just from seeing it, you can see that these two processes, these two heart rates, they are different. They are different, and it's probably you can say that one heart rate is actually more deterministic than another one. So if we live in a stochastic world, of course, it would be good to have kind of a measure, kind of a tool to compare different stochastic processes between each other, or compare, let's say, to analyze one stochastic processes, just to see how actually it is stochastic, because some processes, they are more stochastic than the others, and I thought you saw the examples of that. uh, uh, so uh, in the modern science, people suggested such a tool, and this tool is known as a correlation function. How actually to work? I have to say right now that what I will be talking on this particular slide and the next ones, it's a very simple, very oversimplified actual explanation of correlation function. And those of you who are familiar with the formalism of mathematical formalism, please forgive me for this very actually simple introduction to the correlation function. So having time-dependent processes, let's say we have function A that dependent on t, we can plot the correlation function by actually taking the sum of the products of this function A at different times. So this mathematical notation represents the correlation function. And if we will sum these products of the value of function A at time t with the value of function A at time t plus tau, we can calculate how actually the value of A, function A, correlates with each other with the time distance of tau, right? And we can do the same, we can calculate the sum of these products for many actually points. And the similar approach we can make if we will calculate the correlation function, taking actually much shorter period of time, let's say, as you can see on this example. This is extremely important when we are talking about the dynamic light scattering, because having the values of this correlation function for different taus, for shorter tau time distance or for a larger period of time, we can analyze the scattering from solution. And the rule number one that you guys should take with you at the home is that dynamic light scattering. There are, by the way, there are three different names for this method: dynamic light scattering, quasi-elastic light scattering, photon correlation spectroscopy. But in general, dynamic light scattering experiment is measuring stochasticity. It measures how kind of time-dependent function, like scattering intensity, is correlated over a different period of times. So just to understand how it works, I would like to present here a general light scattering experiment. Here we have a source of light. This is a laser. It goes through the lens to the sample. That could be a solution of proteins. And the scattered light, the light that is actually deviates from its original path, is gathered by detector, and then it's analyzed by a device that is known as correlator. So let's just make a hypothetical experiment. Let's take two different proteins with very distinct molecular weights, let's say 5,000 and 500,000 Daltons. And we will make the same experiment, we will make light scattering experiment from these two solutions. And from the analysis of the intensity of the scattered light as a function of time, you can see actually two issues. There are two significant issues that we can actually say right away. First, the scattered light fluctuates. You can see that for solution A and for solution B, the scattered light actually is a very stochastic. It's a very random value. And another issue that you can see probably that these fluctuations, they depend on the particle size. So for the solution of a larger protein, these fluctuations, they are not so actually random. There are some areas where the intensity is not jumping up and down, but it actually behaves quite deterministic way. So now I would like to, I will try to explain actually why scattered intensity fluctuates and why these fluctuates depend on the particle size. So first I will address to the intensity of the scattered light. First of all, what you have to remember that intensity of the scattered light is proportional to the number of particles in a scattered volume, right? So the more particles we have in solution, the higher will be the value of the scattered intensity. So I think this is very actually straightforward. And another issue that this number of the that we actually the number of molecules, number of proteins in solution that we are measuring is there. It's fluctuating because of Brownian motion. So any nano-size object that we have in a vial will be actually in motion because of Brownian motion that was actually discovered a couple of centuries ago. So uh, when we are making our light scattering experiments, we have a vial here, and actually we are gathering information not from a whole vial, but from a volume that is known as a scattering volume. And this volume has no solid walls. So this volume has opportunity for the outstanding for surrounding particles to come in and come out, and that's why actually the number of particles in this scattering volume that is actually determined by a laser and detector is changing. So some every time we have some number of proteins in a scattering volume, and this number is changing because some proteins they are leaving the scattering volume, and some proteins they actually coming in. Okay, I think this is pretty clear. And what I would like to do with your help, because I need your imagination, I would like to check this scattering volume, and we will be actually taking a picture. We will be taking a picture using this old-fashioned camera, just to see how the number of these proteins is changing with the different period of time between these snapshots. So you can imagine this old-fashioned camera. We will make a snapshots, and every snapshot will have a time, kind of delay time or lag time between snapshots, and we will be actually increasing the time, this delay time, starting from very short period of time, like 0.1 microseconds, to very actually long times, up to maybe 1 second or 10 seconds, right? So here, the blue color stands for the molecules, for the proteins that are leaving from us. They actually goes far from us, and the red ones they actually approaching to us. As you can see here, there are no solid walls. Some of them, some of particles can come in, and some of the particles cannot come out. Well, if you have problems with this the model, you can imagine some another situation. Let's imagine you have a common room full of drunk people, drunk students, and this common room has many doors, and every 10 minutes somebody is coming in, coming out. So basically, we will be trying to make a picture of these drunk people, and with some period of time, right? Okay. Coming back to the proteins in solution, let's start with a very short period of time between snapshots, and that could be 0.1 microsecond. Uh, the question is now, will be there will be any changes with this short period of time between snapshots? It's hardly, right? Because it's the time is definitely not enough for to move. Well, I can provide here some calculations based on the diffusion coefficient, because the Brownian motion is controlled, is governed by diffusion coefficient. So with 0.1 microsecond delay time between snapshots, there will be no changes between snapshots, and that means we can actually predict what how many students or how many proteins will be on the next snapshot, and that means that we have a very deterministic, a very predictable system. We know exactly how many proteins we will have in every 0.1 microsecond. And if we will calculate the correlation function that I presented a few slides before, we will get the values something like close to 1. That means that a probability to predict the number of drunk students in next one 0.1 microsecond is almost 100%, right? Now we will try to make the same experiment, but with a much longer delay time, let's say 10 milliseconds, or speaking about students, let's say 10 hours. Can we predict the number of students, a number of proteins from the previous snapshots? Uh, I guess we can't, because during this delay time between snapshots, there were so many events when some molecules came in, some molecules came out. So the value of the number of proteins in the next snapshot will be almost uncertain. And probability to predict this number is almost zero, right? So for such long uh, lag times, such long delay time, we will have no correlation. But selecting some intermediate delay time, we will probably have the probability to predict something like 50%. And the concept of dynamic light scattering says that this characteristic delay time between snapshots, which is equals 50%, uh, is directly related to the diffusion coefficient, right? That's how dynamic light scattering works. It measures, it measures the fluctuations of intensity of scattered light, it calculates the correlation function, and then by selecting different lag times, different time between snapshots, it's just measuring the time dependent of correlation function. And from this analysis, from its behavior, you can get the value of a diffusion coefficient, right? So coming back to the physics, this is the directly comes from this diffusion law, right? So the delta x squared is proportional to diffusion coefficient. So this random motion of particles who that are making Brownian motion is directly proportional to the diffusion coefficient. Okay, and the rule number two that I would like to provide you is, and this is a take-home message, that dynamic light scattering in diluted solution of non-interacting monodisperse nanoparticles gives nothing else but a diffusion coefficient, right? It is alpha and omega of your dynamic light scattering experiment. Everything else that you probably get from your instrument is based on assumptions. Uh, in general, dynamic light scattering measures, calculates the diffusion coefficient. So let's just have another brief look on the typical experiment. So we have the time dependence of the scattered intensity, and we have solution of two types of particles, solution of big particles, solution of small particles. They are clearly different in terms of fluctuations. The correlator, the hardware, calculates the correlation function, and the correlation function is changing from very deterministic to very stochastic one, right? And depending on its behavior, we can calculate the characteristic time, and we can calculate the diffusion coefficient. And it works in the following way. Um, so for monodispersed particles, the correlation function is supposed to be the exponential function. And from the analysis of exponential function, we can get the characteristic time. And this characteristic time, tau, is directly proportional to diffusion coefficient. Right. Now, I hope I convinced you that using dynamic light scattering from the solution of monodisperse, non-interacting dilute proteins, food proteins, we can get experimental value of diffusion coefficient. But, well, we definitely, we would like to know something else, right? Probably the size of our species, what kind of, well, object we're dealing with. And the first question on this answer was given by Sir George Gabriel Stokes, who was making experiments with some microscopic spheres that were actually falling down through some oils. So he was making experiments with the different sizes of these microscopic spheres with the viscosity of oils, and his experiment were actually generalized mathematically explained by this guy. I think you know him. And this equation is known as a Stokes-Einstein equation that actually tells you that the radius of a hard sphere, because Stokes was making experiments with the hard spheres, is reversely proportional to the diffusion coefficient. So the lower, the smaller nanoparticles, the smaller proteins we're dealing with, the higher will be diffusion coefficient. And reverse, the bigger nanoparticles, the bigger emulsions we're dealing with, the bigger proteins, the lower will be the value of the diffusion coefficients. And to make these calculations, you have to know the temperature, because temperature definitely somehow influences on the diffusion, and the viscosity of a solvent. So the experiment, a classical experiment, works in that way, that we are actually measuring the correlation function. So this is again to remind you, at short delay times we have very deterministic the situation. Here we have a very probabilistic or stochastic, and from the analysis of this the correlation function, we are getting the diffusion coefficient, and from diffusion coefficient we calculate the so known the hydrodynamical radius. And this hydrodynamic radius is the value of a hard sphere. That's what you should remember. This is the value of hard sphere that has this diffusion coefficient. But in general, this is an assumption. From dynamic light scattering experiment, we have just a diffusion coefficient. And the software calculates, it gives you the value of RH, assuming this is a hard sphere. That's probably not true for your particular sample. Okay, now I will explain, I will try to convince you that dynamic light scattering is very useful, especially for food science and nutrition science, because any nanoscale objects could be measured by dynamic light scattering. So the proteins, any proteins in solution, is actually a very convenient and very interesting object for dynamic light scattering experiments. Any polymers, including carbohydrate polymers like starch, dextran, whatever, could be also studied by dynamic light scattering. They have a nanosize range. They are in a nanosize range, and dynamic light scattering can give you the value of diffusion coefficient of these particular polymers. And of course, emulsions also could be studied by dynamic light scattering. So just to give you an idea how it works from the hardware point of view, I presented here the picture of the fluctuation of the scattered light, and the device that is known as a correlator is splitting this the value of the intensity into different. It chops actually the scattered intensity, and taking the value of scattered intensity, and then it put into different segments, different memory cells, and then is calculating during the real time, during the experiment time, calculating the values of correlation function for different delay times. And that's how actually it's supposed to work, and how for diluted monodisperse, well, ideal solution. But in fact, you basically you probably will have such a system maybe once or twice in your life if you're working hard, if you're working with dynamic light scattering. Most of the system actually I was measuring in my life were bad. They were either not non-ideal in any sense of view, from any point of view. So speaking broadly, what could be wrong with my dynamic light scattering data? How actually I misinterpreted the dynamic light scattering? So, and to answer this question, you have to remember the rule number 2. Dynamic light scattering in the diluted solution of non-interacting monodispersed nanoparticles gives nothing else but a diffusion coefficient, right? So it's highly possible that your solution is polydispersed. It might be the case when you have bimodal or polymodal distribution. You have two types of particles, let's say proteins in their aggregates. This is a classical example of what you might have in your vial. It's also possible that concentration of your proteins is very high, and you can't regard anymore the solution as a diluted. And your particles, your proteins, your emulsions could be charged. So the charges also brings additional difficulty to the interpretation of dynamic light scattering. So all three possible situations, they make life very complicated from the point of view of interpretation of dynamic light scattering. And the rule number 3 that I would like to also explain you is that dynamic light scattering is a blind method, right? Why? It's because it gives you only the value of a dynamic light scattering, a value of the diffusion coefficient from the dynamic light scattering. And then you program will convert it from correlation times, from the characteristic time to the hydrodynamical radius. But again, you never know if you are dealing with a polymeric coil or silica particles or lysozyme or dendrimer or glycogen or tobacco mosaic virus. The only value you will have from DLS experiment, it's just diffusion. That will be converted by software to hydrodynamic radius. But it doesn't mean that your particles are spheres, or they could be actually, they might have different shapes, like I presented here. So what to do? Well, of course, the best way how to address to this question about the shape of nanoparticles is to make cryo-TM experiments or AFM experiments. But if you don't have access to this facility, there are some tricks, however, that you can use for the analysis of dynamic light scattering. And I will try to give you uh, some explanations how these tricks can be used. So the first trick is actually works on the analysis of the scaling exponent. Let's assume that you synthesized polymers in a broad range of molecular weight. So you have polymers or proteins, whatever, with the similar proteins, the same protein, with the range of 5,000, 10,000, 500,000, or 1 million Dalton. Or it could be, let's say, kind of rod-like structure. And then if you will measure the hydrodynamic radius for this particular object and plot the behavior, the dependence of this hydrodynamic radius as a function of molecular weight, you will get the value of a scaling exponent. And this scaling exponent is very sensitive to a particle structure. So for hard spheres, remember that volume is proportional to the radius power 3. So for hard spheres, the scaling exponent is 0.33. For rods, it's alpha equals 1. And for polymer coils, depending on the quality of the solvent, alpha could be in the range of 0.5, 0.6. So let's just apply this trick for very interesting object, for glycogen. So glycogen, it's a branched polysaccharide structure that is basically the storage of energy. Glycogen helps us to do sport, to make some physical activities. Every human being has something like 200, 300 grams of glycogen. And the runners who are running a marathon, they actually know that once we have glycogen, we can run faster. But when we are running out of glycogen, the body starts actually digesting fats, and this is actually is known as a marathon wall. in my work, something like 13 years ago, we have selected glycogen of different source. We have taken from sigma bovine glycogen, blue mussel glycogen, rabbit's glycogen, oyster, because glycogen actually is a part of a variety of animals, including some bacteria and fungi. So when we measured the hydrodynamic radius and plotted as a function of molecular weight that was actually known from literature, we received the dependence of the hydrodynamic radius as a function of molecular weight, power of with the scaling exponent 0.33, that immediately tells us that the glycogen is a sphere, right? So that's how actually you can analyze the structure of your object using only dynamic light scattering. Again, to make such an analysis, you must have a series of similar objects with a broad range of molecular weights, right? Most probably, you don't have you're not so lucky to actually to make it. So, well, I guess you have to make some kind of microscopy experiments to get an idea about the shape and maybe the internal structure of nanoparticles. So another issue that you have to remember when we're talking about blindness of dynamic light scattering, that intensity of scattered light, remember, it's proportional to the number of particles, but it also depends on the size of particles. The bigger particles scatter much stronger than smaller particles. For hard spheres, the intensity is proportional to the sixth power of a hard sphere, right? So let's just make again a hypothetical experiment. We will uh, we will measure the mixture of two proteins. One protein will have the size of 5 nanometers, and another protein will have the size of 15 nanometers, right? So, and in solution, we will make them with the same concentration. So their molar ratio will be equal. So they actually will be 1 to 1. But making dynamic light scattering experiments, measuring the scattering intensity, we will end up something like that. So that's you can see that the program that is providing the analyzing dynamic light scattering will show you actually the two peaks, and one peak is much actually stronger than another one. People call it actually it happens because the dynamic light scattering measures scattered intensity, and scattered intensity is proportional to the sixth power of radius. And the 15 nanometers proteins scatters 1 million times actually higher than 5 nanometers experiments, the proteins. And again, that's what you have to keep in mind when you will be analyzing your proteins, your food samples by such experiments, such a method as dynamic light scattering. Uh, so uh, it's also possible that your particles, your proteins or your emulsions, they are polydisperse, and dynamic light scattering experiment provides the distribution of hydrodynamic radius, and it has three numbers. One number is based on intensity, and it's known as intensity weighted. Another number is known as a volume weighted, and third number is known as a number weighted. So the experimental value that you are getting from the DLS is the intensity weighted. Another two, they are based on some assumption that your particles are spheres, and actually it's provided for users just to estimate the size, assuming that you have actually to get rid of the scattering, to somehow suppress this radius power 6 behavior, right? And many users, many students, they're actually confused, so they're measuring their actually maybe glycogen or whatever proteins, and they see three different numbers. For example, for oyster glycogen, you can get from the software 25 nanometers, 15 nanometers, and 12 nanometers. So the question one, which distribution is better, right? Well, both values they are right, because you're actually working with a polydispersed system. You have certainly you have some particles with the size of 25, with the size of 15, and the size of 12. Many times people observed a certain discrepancy between the value of the hydrodynamic radius that is provided from dynamic light scattering from the values provided by microscopy from cryo-TM, for example, microscopy experiments. And in many papers, there was a discussion of what actually the source of this discrepancy. Well, actually, the main reason for this discrepancy comes from this different weighting procedure. Dynamic light scattering, the first value that you have is based on scattered intensity. The biggest particles that are scattered the most, they are somehow the mostly represented in your actually in this distribution. Whereas the transmission electron microscopy is based on number weighted distribution. And that was nicely demonstrated by a group of French scientists. So they measured nanoparticles that are formed from polymers, and they measured the intensity average diameter for a variety of nanoparticles, and then they converted to number average diameter. And also they compare it with the average diameter from the transmission electron microscopy. So you can see here that intensity average diameter is much higher than number averaged, and it's higher than the diameter from transmission electron microscopy. But if you will convert intensity average diameter to number average, you see actually quite nice agreement between these two values, and that's how actually you have to do if you are really concerned about this discrepancy about microscopy and the dynamic light scattering experiments. Just remember that dynamic light scattering is based on the scattering intensity, right? So it's based on the scattering, and the bigger particles scatter actually much higher than smaller particles. So a very important issue is the resolution. Let's just assume that you have three types of nanoparticles in solution, three types of proteins, right? Can you resolve these proteins from your dynamic light scattering data? Well, actually, if the ratio between these diameters, the sizes, is less than 3, you can't do it using by dynamic light scattering. From this paper published by Australian team, they have taken the latex particles with the size of 220, 330, and 410 nanometers. They mixed them actually together, and they analyzed it separately by a bunch of experimental methods, such as transmission electron microscopy, dynamic light scattering, particle tracking analysis. So you can see that the TEM can visualize these particles independently and can analyze them when they are mixed in solution. However, dynamic light scattering can give the value of hydrodynamic radius when you have just one type of nanoparticle solution, but then mixed, it just gives a very broad peak. So it's possible that instead of bimodal distribution, let's say you have mixture of protein 1 and protein 2, if the size, if the radius is just not very far from each other, you will see just a broad peak instead of actually two peaks on the distribution function. This is another actual source of a problem. So we will keep going with the problems of dynamic light scattering that you can get. Again, I told you already that dynamic light scattering is measuring the diffusion coefficient. But if you're working with a very complex system, with a very concentrated one, if you are working with the charged particles, it's highly possible that you might see some artifacts, or alternatively, you might not see something that exists in solution. So the rule number three, not all peaks on distribution functions are particles, and not all particles show a peak on distribution function. Yeah, for example, if you will take a polymer and you will measure it in a bulk without any solvent, and you will put it in dynamic light scattering instrument, you will also see some kind of correlation, right? And this correlation will be dependent on temperature. And so this in this case, you have not a solution, you have just a bulk material. And inside of this bulk material, you have some kind of thermal motion. So dynamic light scattering will show you correlation, right? It will show you some non-zero correlation function. But I guess if you are working with the solution of proteins, you shouldn't concern about this very much. But again, there are no particles here, but we do see some kind of changes of correlation function with time. And if we will calculate it distribution, you will see some peaks, but these peaks are not real. They just show it shows you that we have some kind of thermal motion in this bulk material. Another situation when we have, for example, proteins, but we don't see them on dynamic light scattering, is when the refractive index of your protein is matched to refractive index of a solvent. For food science, it's not a very actually common situation because mostly you will be dealing with water as a solvent. But if you are working, let's say, with the polymer science, if you, let's say, will be use a toluene or some organic solvents with a much higher refractive index, it's possible that the scattering from your object, from your nanosize object, will be actually equal to the scattering of the solvent. And that is known as a isorefractive point. When I was working for the first time with some polystyrene latexes, and I used toluene as a solvent, actually I was very confused because my correlation function was almost zero. And the reason for that, because the refractive index of solvent toluene was actually perfectly matched to the correlation to the refractive index of latex. So again, it's possible that you, even if you have some kind of nice particles in solution, you will see anything. And this is a clear example, a manifestation of isorefractive point. Okay, another actually important issue that might happen to you is that you have, again, bimodal population in solution. You have, let's say, proteins, and you have aggregates. Some proteins, they do aggregate. And if these aggregates, they're very large, it's highly possible that you will see no actually single proteins, but you will see just aggregates. And that actually happens again because of this r power 6 dependence. So your large objects, large aggregates, several hundred nanometers, they just outnumber it, the scattering from your single small stuff. And it happened to my students, it happened to me many times. So people are coming to me with their samples, and they just measuring by dynamic light scattering and say, look, it's weird. I'm pretty sure that I synthesized some nice particles. They should be the size of 5-10 nanometers, but I don't see them. I see just something like 500 nanometers. And what's going on? Is something wrong with your instrument? But actually, there's nothing wrong with the instrument. It's just because you should get rid of these large entities in solution, and only after that you will be able to measure the hydrodynamic radius of your small proteins, small nanoparticles. Okay, and the final probably example of some uncertainty, of some problem that you might experience with the analysis of dynamic light scattering is when you're working with concentrated solution. because diffusion coefficient that you are measuring depends on concentration of your proteins, depends on concentration of your emulsions. And this happens because if there are so many particles together in solution, they will start bothering to each other. So their diffusion is not diffusion of a single molecule, a single protein, but they actually prevents they just bother each other, and their diffusion coefficient will be changed, right? Um, just imagine that you are a runner, and you have to run from one through the square full of tourists, right? If there's nobody here, so your pace will be your real pace. But if you are supposed to run in a crowd place, like for example in this square, of course your pace will be severely, let's say, influenced by some surrounding people. And actually, it's the same with diffusion. So if you have very concentrated solution above C star, you can't measure the real diffusion coefficient, but you're measuring so-called apparent diffusion coefficient, right? And just to get the real diffusion coefficient, the true diffusion coefficient, uh, you have to measure a series of concentrations, and then extrapolate this dependence of the diffusion coefficient to the infinite dilution here. And from that, you will get an idea about the true diffusion coefficient, or and then after that, you can actually calculate the true hydrodynamic radius. Well, yeah, so just to give you over an idea how it might actually look like with working with some carbohydrates, with the polysaccharides. So this is the paper published quite a long time ago. Uh, Japanese scientists, they studied the polylan. It is a nice polysaccharide polymer, and the polylan was had different molecular weights: 50 kilodalton, 100, 200, and 500 kilodalton. So they measured the diffusion coefficient as a function of concentration of polylan in solution, and you can see that this diffusion coefficient is clearly concentration dependent. And again, some people might be confused with that. Uh, the software provides to the users the value of hydrodynamic radius, and they actually measuring the series of concentration, and they see that the hydrodynamic radius is changing, and they are saying, "Oh, look, my particles, they are getting bigger or smaller, so I have some kind of a new phenomenon." But this is not true. What actually they observed is just the concentration dependence of diffusion coefficient, right? And it's not related to the size of nanoparticles. What you have to do to make the correct value, you have to measure several concentrations to measure diffusion coefficient, and then extrapolate it to infinite dilution. And from that, you can get the true diffusion coefficient, and from Stokes-Einstein equation, you can get the real hydrodynamic radius. Okay. Okay, so some hints. How much time actually do I have? Martin?
I think, well, it's 8 minutes to two
Uh,
now. I'd be good to wrap
okay, I will
up.
I will do it very fast.
Sure.
Okay, so just how to measure decent DLS data. First, you have to prepare a decent sample. You should start with the sample preparation. Then you should inspect the count rate. Then you should inspect a correlation function, and then you should inspect a distribution function. Okay, so again, dynamic light scattering measures in scattered intensity, right? And if you have dust in your solution, this is highly probable. These dust particles, they're very big. They are of micron size, and they will spoil your experiments. And that's why you have to filtrate your solution. If you In other words, dynamic light scattering is very sensitive to dust. If you didn't filtrate it properly, you probably will see on the count rate, that is a dependence of intensity, scattered intensity is a function of time, you will see some spikes. And these spikes, they come from dust, right? So by default, I'm taking a filter, PVDF or some other hydrophilic filters, and I'm filtrating it to the cuvette. And if you don't know which filter to take, there are many charts available from different companies. If you're working with organic solvents, this chart will tell you if your particular filter, PTFE, PVDF, is actually robust against of a particular solvent, right? But if you're working with the aqueous solution, I would recommend you to take a PVDF or PES filters. They're very nice and very hydrophilic filters to take. So working with filters, always remember that filters could come in a box. They could be they could be in a special kind of envelope, or they could come just without any this just in a box. So I would recommend you to rinse yourself with the filtrated water first. Again, if you before the filtration, the real solution, take off first 2-3 droplets, because dust could be inside of a filter as well. So then, as a step number 2, filtrate your solution in a dust-free cell. Again, if you're using another filter, take off first 3 droplets. And also remember, if you're working with a low volume, that some amount of solution will stay inside of a filter, right? So, and then again, check refractive index if you're actually suspicious that if some amount of material was retained by a filter, if you have some kind of interaction between filter material and your maybe proteins. Okay, then select a appropriate cell. That could be plastic, it could be quartz cell. They are all available. You can take just a spectra photometry cell, or you can take some plastic cells. But remember, the thicker the cell you're working with, the better you will have the correlation function, because you are gathering information, you're gathering the scattered light from the actually bigger volume. And then so the step number 2 is inspect your count rate. So this is a snapshot from Malvern software. So this is the correlation function. It is a nice and a single exponential, and this is the count rate. Okay, so this is a good count rate, but it's possible that you will see some kind of spikes, and this is actually not a very good count rate. And this count rate is just awful, right? If you have something like that, you should stop your experiment and maybe start thinking what's wrong with my solvent, with my solution. Okay, then if you are happy with your count rate, just inspect your correlation function. In general, for monodisperse solution, you must have similar like this. It will go from something like 0.95 to almost to 0. And it's possible that in the presence of dust, you might see a background. And again, this is the point of start thinking about getting rid of some maybe dust that somehow is a part of your solution. Or if you have this correlation function, this is just a tragedy. Again, don't try to analyze it. Don't try to read the value that software will provide you, because it's just just a software, right? It will give you some numbers, but these numbers of hydrodynamic radius, they are just bad. They are not real. Well, program is can calculate everything, right? But these data are not reliable at all. Of course, Malvern software provides also pole dispersity index, and if it's above 0.5, probably you are working with the very pole disperse samples. Then inspect your distribution function. It's possible that you have bimodal distribution. And if in this case you should check the data, additional data you can see in these folders where you will get the values of hydrodynamic radius for every peak. So by default, the program gives z average, a very averaged number that is average over two peaks. So if sample is dust-free, use longer measurements and just one run. This measurement can be let's say 100 seconds, 50 seconds, 150, 200 seconds. Well, if you have some dust, use multiple runs and shorter measurements. Always check the count rate, and yeah, just check the quality report. And these are the take-home messages, right? So I think I should stop at this point, and thank you for your attention. Sorry for being long.
Okay, thank you
Martin?
very much. That's a really interesting talk. I don't know if anyone has any quick questions. Feel free just to put your hand up and ask away, or write it in the chat. I will ask just from a purely practical perspective. So do we have the equipment available at the university? Is this something in CAF or in your own lab?
Yeah, there is a Malvern instrument in the LG3. It's Vitalis Khutaryansky lab. So I think some people from food and nutrition science department, they usually come and measure on this instrument. So it's available for users.
Okay, great. And another quick question. So it strikes me that this requires some fairly pure samples. I know a lot of sort of food samples clearly are quite complex mixes of different chemical compounds and unknowns. So would I be right in saying that you would need a fairly strict purification step before you start work?
Yeah, purification is very crucial for dynamic light scattering, because if you will grab your sample non-purified and will measure it on dynamic light scattering, you will have a very broad distribution function with the multiple peaks, and this is almost impossible to analyze. It's just it's very polydispersed. So basically, you have to filtrate it, or maybe you have to split on different components if possible. Yeah. So unfortunately, dynamic light scattering is very sensitive to polydispersity, to some kind of complex structure of what you actually are measuring. If you have several types of particles, if you have, let's say, large and small emulsions, if you have whatever, it's polydispersed. Actually, it's a big problem.
Okay, yeah, I can certainly see the utility though in terms of looking at protein structures. Tannins are something that sort of jumped to mind, and melanoidins, sort of some of these larger color compounds, which are larger polymers, I suppose. So does anyone have any questions from the chat?
I think people were tired by my long presentation. Yeah, sorry for that. Usually
right.
I'm given a full day seminar workshop about it. So it's very hard to talk about dynamic light scattering just in 15 minutes.
It's clearly a complicated procedure. There is a question from Roman. Thank you for such useful talk. Some researchers present size data in nanometers in their papers as whole numbers, while others put at least one number after the comma. Is there any strict rule in terms of this? According to the Malvern website, nanosizer allows to measure size particles as low as 0.3 nanometers.
Okay, yeah. Thank you, Roman. Again, in general, the dynamic light scattering for monodisperse system can measure hydrodynamic radius with the accuracy of 0.3 nanometers. It's true, but there is another source of errors, right? if you make an experiment several times, you will end up with a bit different hydrodynamical radius. So it will be, you have to average your RH value over several repetitions, and this repetition error will be actually usually is much higher than the error of the one single experiment, right? So it, it depends on the system, but it's true that in for very nice, very dispersed system, you can get RH value with accuracy 0.3 nanometer. It's possible, but in reality, the accuracy could be plus minus 1 nanometer, maybe even higher. Depends actually how good your system is, how your system is good. You have to just make several repetitions, several runs, and then calculate the accuracy depending of this, well, your fluctuation of RH.
Okay. Well, I hope that answers your question. In that case, feel free to get in touch. I think we have. Oh no, we do have a hand up. Sorry, yeah. Sultan.
Hello.
Hello.
Thank you, Sergey, for your presentation. We are working on right now dark field light scattering, and we are trying to detect the bacteria and bacteriophage effects on the bacteria. So, and we realized that our system, we build actually a dark field light scattering system, which is a really basic model, but we just realized that we can also measure the platelet aggregation on the system. So I just wonder, do you have any, did you do any research for the bacteria detection, light scattering for bacteria, or the platelets work?
Well, dynamic light scattering usually works with solutions, right? So if the upper limit for DLS is something like few microns, one maybe 3 micron, and bacteria, I guess they are usually larger than this upper limit. So as I said in the beginning, the crucial thing is to have Brownian motion. And if you have larger particles, and let's say some kind of swimming particles, it's not a Brownian motion anymore. It's something else. If you have large particles, they will sediment on the bottom, and it's very hard to analyze it by dynamic light scattering because it's just a sedimentation. It's and if you have bacteria and they whatever. I guess it's not a good system for dynamic light scattering. The bacteria, they are too big, probably. I never heard about it.
The thing is, the bacteria which we use, they are not motile, so they are not moving around.
Okay.
Even in a liquid. So.
But how big are they?
Yes, pretty big.
Yeah.
200 micrometers.
Okay, so just if you will take 200 micrometers size microparticles, you will see clearly precipitation in solution, because Brownian motion is not enough to keep them in solution. They will will go down, and instead of nice decay of a correlation function, you will see some oscillations like that. I guess not. You can try maybe master size. Yeah, I think you need a master size that based on Fraunhofer diffraction. If you're familiar with this method, I think it's available somewhere in food and nutrition science. So instead of making dynamic light scattering, you can measure a total scattering as a function of angle from solution, and it works very good for large particles. So you just write me email, and I can provide you more information about
Thank you
Fraunhofer
so much.
diffraction.
Actually,
Yes.
a paper to compare. We actually prove it works for the bacteria, and we published it. And now we just after realize it works for platelet aggregation, but it's really hard to say measurement of the platelet aggregation because they are quite small, and we just confused to understand it. So yes, it will be really nice to contact with you, and we have also some data. Maybe if you want, you can check.
Okay.
Just to check. And okay,
I will do my best if I can help. Sure. Well, I will type in the chat my email. Just.
okay.
But again, the dynamic light scattering works with nanoparticles, something from 1 up to the 1 nanometer up to 1 micron. Well, it's not micro-sized particles is not a good system for DLS.
Yes, I see. Thank you, by the way.
Okay. So this is my email. Just drop me a line if you want to talk.
I got it. Thank you so much. Thank you. It was nice talk.
Thank you.
Okay, well, thank you all very much. I'm going to have to wrap it up here because I've got another meeting to get to. So yeah, feel free to get in touch with the email in the chat there if you have any more questions. I'm sure Sergej will be quite happy to help. And yeah, thank you very much for the time and the interesting talk.
Thank you, Martin, and thank you for invitation.
Quite all right.
Bye.